Hi, this is Pratima and you are watching Planet Physiology. Today we shall have an introductory session on endocrinology. Endocrinology is study of endocrine systems in human body, which includes endocrine glands, their secretions and diseases related to them. Endocrine glands are glands without ducts, that is ductless glands, which pour their secretion directly into the blood. The secretion is called as hormone. The word hormone is derived from the Greek word hormone, which means setting in motion. The term hormone was coined by Bayliss and Starling in 1902 when they discovered secretin. In 1849, Arnold Barthon realized presence of some chemicals from testes to regulate reproductive behavior in roosters. Oliver and Schaeffer published a full article in 1895 stating adrenal extract contains adrenaline, the substance causing physiological changes. And this was actually the first hormone to be discovered, even though many give credit to the Starling for discovery of secretin in 1902 as the first hormone to be discovered. Starling has defined hormone as a chemical substance produced by an organ and released in blood and transported to distant organs to exert its specific functions. Most of the classical hormones perfectly fit into this definition. So let us start with these hormones and their endocrine sources. The first major endocrine gland is pituitary gland, located in the brain. It has two parts, anterior and posterior. Anterior pituitary secretes growth hormone, thyroid stimulating hormone, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone and prolactin, while posterior pituitary secretes two hormones, antidiuretic hormone, which is also called as vasopressin, and the second is oxytocin. The next gland is thyroid gland, located in the neck. It secretes T3 and T4, which are concerned with general metabolism and various systemic actions. Another hormone secreted by thyroid gland is calcitonin, which is related to calcium homeostasis. Located on posterior aspect of thyroid gland are two pairs of parathyroid gland. These are indicated by these yellow spots in this picture. They secrete parathormone, which is also related with calcium metabolism. Pancreas in the abdomen is endocrine as well as exocrine organ. Endocrine part of the pancreas is in the form of islets of Langerhans, and these islets secrete hormones like insulin, glucagon, somatostatin and pancreatic polypeptide. Located on superior aspect of kidneys is adrenal or suprarenal gland. Outer part of the adrenal gland is called as adrenal cortex, and it secretes steroid hormones like mineralocorticoids, glucocorticoids and adrenal androgens. Inner part of the adrenal gland is called as adrenal medulla, and it secretes catecholamines like adrenaline, noradrenaline and dopamine, and it is integral part of sympathetic nervous system. Primary reproductive organs, that is gonads, also act as endocrine organs. Ovaries in case of females secrete primarily estrogen and progesterone. They also secrete other hormones like inhibin and relaxin. Testes in males secrete testosterone, dihydrotestosterone and androstenedione. Apart from these glands, hypothalamus, pineal gland, heart, kidneys, gastrointestinal tract and many other tissues also act as endocrine organs. Hypothalamus secretes various releasing and inhibitory hormones that regulate activity of other endocrine glands. Pineal gland secretes melatonin, which plays important role in circadian rhythm. Heart secretes atrial natriuretic peptide. Kidneys release renin, erythropoietin, 1,25-dihydroxycholecalciferol and prostaglandins. Thymus is concerned with secretion of thymic hormones, and gastrointestinal tract secretes various gastrointestinal hormones. Adipocytes also are endocrine organs which secrete leptin and other hormones. Placenta secretes human chorionic gonadotropin, chorionic somatomamotropin, estrogen, and progesterone. Now let us study how to classify hormones. On the basis of their chemical structure, hormones are classified into three major groups: proteins or peptide, amino acid derivatives, and steroid hormones. Protein or peptide hormones are made of peptide chain containing variable number of amino acids. They may be oligopeptides containing less than 10 amino acids like antidiuretic hormone and oxytocin, or they may be polypeptide with more than 10 amino acids, for example growth hormone or insulin. Based on their structure and functions, these hormones are further subclassified into different families like insulin family, growth hormone family, secretin family, and glycoproteins. The second group of hormone is amino acid derivatives. As name suggests, these hormones are derived from amino acids, most commonly from amino acid tyrosine. They include hormones like epinephrine, thyroxine, and melatonin. These hormones are hydrophilic in nature. The third group is steroid hormones, which are synthesized from cholesterol and include adrenocorticosteroids and gonadal steroids. These are hydrophobic in nature and hence can easily cross the cell membrane. Apart from these three major types, some hormones are synthesized from lipids like arachidonic acid, lipoxin, thromboxane, and prostaglandins. For example, various prostaglandins and thromboxane. These are called as eicosanoids. The enzyme necessary for formation of these hormones include cyclooxygenases and lipoxygenases. Eicosanoids are hydrophobic in nature and they act through membrane receptors. And the last category is gases like ethylene and nitric oxide. Based on the function, hormones can be classified as releasing hormones, which cause release of other hormones. For example, growth hormone releasing hormone secreted by hypothalamus, which causes release of growth hormone from anterior pituitary. Similarly, corticotropin releasing hormone causes release of ACTH from anterior pituitary. Second category is inhibitory hormones, and they inhibit release of hormone from the endocrine glands. For example, prolactin inhibiting hormone prevents release of prolactin. Growth hormone inhibiting hormone prevents release of growth hormone. Both these types of hormones are secreted from hypothalamus. Hormones can be tropic in nature, that is, they are stimulating hormone. They stimulate the activity of other endocrine glands. For example, adrenocorticotropic hormone, that is ACTH, stimulates adrenal cortex. Similarly, TSH, thyroid stimulating hormone, stimulates thyroid gland. And the last category in this classification is non-tropic hormones, which directly act on tissues. For example, growth hormone, cortisol, or vasopressin. All these hormones act as chemical messengers. So now let us study their mode of communication. Intercellular communication. The first type is endocrine communication, where hormones act on distant tissues by traveling through the blood. Most of the classical hormones like growth hormone, thyroid hormone, parathormone, etc. are included in this category. As hormones are released in bloodstream, they get diluted and are present in smaller quantities. Also, they are slower in action. The second mechanism of intercellular communication is where hormone diffuses into the extracellular fluid and acts on the neighboring tissues. This type of communication is called as paracrine communication. Example is somatostatin from D cells of pancreas acting on the other cells of islets of pancreas, influencing secretion of insulin and glucagon. Also, most of the eicosanoids fall in this category. These chemicals are usually called as local hormones. They are degraded quickly and thus are prevented from entering into the general circulation and spreading to the distant sites. The third category is autocrine communication, where chemicals released from the tissues act on itself. Thus, the hormone affects its parent cell functioning. For example, platelet activating factor is released from platelets and acts on themselves. Another way of intercellular communication is by neuroendocrine mechanism. The secretion by the neuroendocrine cells enter the bloodstream, and the secretion is called as neurohormone. For example, secretion of posterior pituitary gland, ADH and oxytocin. Now let us study how hormones are transported in the blood. Peptide hormones and catecholamines, which are water soluble, do not require any special transport proteins. They are transported in dissolved form in the plasma, and hence they also have less half-life. In contrast, steroid hormones and thyroid hormones are water insoluble and hence are transported with the help of plasma proteins. Hormone in combination of plasma protein is called as bound form of the hormone, and this bound form also acts as reservoir of the hormone. This prevents rapid changes in hormone levels in the plasma. Bound form also acts as inactive pool of the hormone and prevents rapid degradation of hormone, and hence these hormones have more half-life. For example, half-life of thyroid hormone is 6 days, where most of the thyroid hormones are present in bound form. Whereas half-life of aldosterone is just 25 minutes, as small percentage is transported in bound form. Free form of the hormone is biologically active form that binds with receptors on the target tissue and exerts its action. For diagnostic purposes, estimation of free form of the hormone is more important than that of total hormone levels in the blood. And this is because the bound form can be altered in various conditions like pregnancy, and hence determination of total hormone may be normal, but the person shows deficiency symptoms due to less amount of free form of the hormone. Now coming to the degradation of hormone, liver and kidneys are major site for degradation of hormones. Most of the hormones are excreted via urine, and hence measurement of urinary concentration of hormone degradation products are useful in assessing their rate of secretion. For example, measurement of vanillyl mandelic acid or VMA in urine is index of catecholamine secretion. Metabolic clearance of hormone is also useful in assessing hormonal dysfunctions. It is calculated by dividing the amount of hormones removed per unit time by the plasma concentration of the hormone. Now coming to the measurement of hormone concentration, the most common method to measure hormone levels in the blood sample is by radioimmunoassay. It is competitive binding assay where radio-labeled hormone and the natural hormone in the blood sample compete with each other for limited number of antibody binding sites. After the equilibrium time, hormone antibody complex is separated and radioactivity is measured. The results are compared with standard curve to find out the hormone concentration in the blood. The major drawback of the technique is it measures immunoreactivity and not the biological activity of the hormone. To overcome this, instead of antibodies, specific receptors for the hormone are used as binding reagent, and now this method is known as radioreceptor assay. It measures biologically active hormones. Sometimes ELISA, that is enzyme-linked immunosorbent assay, also can be used to determine hormone levels in the blood. It is enzyme-based colorimetric or fluorometric assay and does not produce radioactive waste. Before discovery of radioimmunoassay, bioassays were used to determine hormone concentration, where ability of the hormone to produce characteristic biological response was assessed. The amount of hormone necessary to produce the response of the specified magnitude under the standard condition was determined. This was a very slow process and quite expensive method and hence not in use nowadays. So let's have a quick recap of the important points. Hormones are chemical messengers secreted from endocrine glands and act on distant target tissues. Based on chemical nature, they can be proteins or peptide hormones, derivatives of amino acid or steroids. Functionally, hormones can be releasing or inhibitory hormones, tropic or non-tropic hormones. Water-soluble hormones are transported in dissolved form and has less half-life, whereas lipid-soluble hormones are transported with the help of plasma proteins and has more half-life. Apart from classic endocrine communication, local hormones act on the nearby tissues by paracrine mode and also on the parent cells themselves by autocrine mode. Secretion of neuroendocrine cells is called as neurohormone, and the hormone levels are measured by radioimmunoassay or radioreceptor assay, and measurement of free form is preferred over total hormones. That's all for today. Thank you for watching and see you in the next session. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.